Hello and welcome to another episode of the Omics Cast. Today we're going to talk about precancerous mutations and cancer detection. And this episode is in response to a couple of tweets that we've seen recently from two people, Eric Topol and Ewan Burney. Eric Topol, very active on Twitter, comes from the medical side of things, and Ewan Burney, also very active on Twitter, comes from the biome side of things. Eric Topol highlighted a couple of recent papers from the GetGuts lab at Broad Institute and collaborators showing this that he calls somatic mosaics, these mutational clones that we all have in our body. And Ewan Burnley replied, "This is why early prediction of cancer is going to be hard. We all will have cancer or precancerous cells, and then the question is, not do these cells exist, about measuring them by a cfDNA, but is our body adequately managing the cancers it has got in it?" And so that's a far more complex system-wide question. And so let's dig deeper into this and see what this is about. So we all have what we call age mosaicism in our bodies. As we age, we accumulate mutations in some of the cells in our body, not all of them. So our body turns into a mosaic of nuclei, where not all the DNA in each of the nuclei is the same. It's a mosaic. And so why does this matter for cancer diagnosis? Well, or for prediction of cancer, because if we have these mutants and we use mutations to measure the probability of someone having cancer, then having mutations in normal tissues is a problem. That is a problem that can be confounding the result. And so what they did in these papers essentially is a sequence, not the DNA, but the RNA that is encoded from the DNA of cells. If they sequence it on normal cells, then the RNA molecules they sequence are going to be normal. But if they sequence it in cells that have somatic mutations, they will see those mutations in the RNA molecules. And so what they've done is that they've done this into multiple tissues in several individuals, and they've measured or assessed which are the tissues with the highest incidence of these somatic mutations. And the highest incidence has come from esophagus, lung, and skin, and in women as well, to some degree in breast, but to the largest degree esophagus, lung, and skin. Skin that has been sun exposed, not skin everywhere. And so this is a perspective from Christian Tomasetti that basically says mutated clones are the new normal. They are everywhere. We cannot get away from seeing them in the body. And then the question is, how is this going to affect cancer prediction? And so the paper by Karen Yitzhak and co-authors looks at this in detail. And what they basically say is that they found a large number of acquired somatic DNA mutations, some of which are typically associated with cancer, and mutational clones of macroscopic size, meaning there's quite a few cells there, in the absence of cancer pathology, meaning the individual is healthy, the individual does not have cancer, but it's got cells in the body that have the same mutations you would find in cancer. Now, these somatic mutations were detected in nearly all individuals and across many normal human tissues in genomic regions called cancer hotspots or in genes that play a role in cancer. So this is a way to say that these mutations are precancerous mutations. They are mutations that when the cancer evolves from not being a cancer, from being precancer into cancer, then they will be hitting the same cancer hotspots or genes. And then interestingly, skin, lung, esophagus are the three tissues with the most mutations, suggesting that the environment generates many human mutations. So skin is probably the easiest to understand, lung as well, especially for people that smoke, and then esophagus because it's affected as well by the environment. So this is something that I learned from a professor in my undergraduate degree which he said essentially that human beings can be described very simply as a cylinder. And here I show this as a toilet paper roll. We're basically exposed on the outside of the cylinder, but we're also exposed on the inside of the cylinder on both ends. So if this end is the mouth, anything that is from the mouth to the other hole in the body, there's also cells that are exposed to the environment, right? And so the easiest example to understand, or in this analogy, is the skin. The skin is the outer part of our cylinder. So skin that is exposed to the sun can accumulate mutations because the UV light in the sun mutates the DNA in our skin cells. How about the lungs? Well, the lungs, when we breathe in and out, we're breathing air that may have components that cause chemical reactions with our lung cells. And so if we breathe in and out dirty air, as smokers do, or from pollution, then we can also accumulate mutations in those cells. And that would be part of the inner part of the cylinder in this analogy. How about the esophagus and the digestive tract? Well, that would be the inner part of our cylinder analogy. So from the mouth here, the esophagus would be on this part, and then at the very end, the colon and the rectum at the other end. And so an example for this is something that has been observed already, which is in some parts of the world, like Iran, they have a tradition, it's part of their culture, to drink scalding hot tea. So they drink tea that is so hot that when it goes down their esophagus, it increases by going down the esophagus the risk of having cancer of the part that is affected by drinking this scalding hot tea, which is the esophagus. So this is a way of explaining essentially why there are some cancers that are exposed to the environment more so than some other cancers. And so the issue here, it becomes, if there are individuals where you identify a large number of microscopic clones carrying somatic mutations, but they are healthy, how far away are we from confidently predicting cancer in individuals that look otherwise healthy? And so a little bit into the details of what they did, they developed this method that they call RNA Mutect, and it was developed during this project called the Cancer Genome Atlas, TCGA, a large multinational project that did a lot of genomics and other omics on cancer. And then what they developed there, they applied to another set of samples that are the GTEx dataset of normal tissues. So these are normal tissues. They are from people that do not have a cancer pathology. What did they find? Well, what they found is that the 3 tissues with the largest number of somatic mutations were sun-exposed skin, esophagus mucosa, and lung. And these findings suggest that the environmental exposure can promote somatic mutacism. And then they checked this with a technique that was developed in the wall of molecular evolution and evolutionary genetics, which is the DNDS ratio, the ratio of non-synonymous to synonymous substitutions. And when they measure it on these cells, this suggests that there's something going on there. And what this is that they say that some of these mutations that they observed may have a selective advantage that could relate to them developing into cancer. So if you have clones of cells in your body that have accumulated certain mutations, and those mutations make them slightly selectively bit better than the cell next to them that doesn't have the mutation, then they can grow a little bit faster, then can grow a little bit bigger, and then they are selectively growing more than the surrounding cells. And they've checked this on bigger mutational profiles like allelic imbalance events at the chromosomal arm level, and they also found those in normal tissues. So again, this is about normal tissues in our body, and then measuring if they have any mutations accumulating, and they found it. And they found it on esophagus, lung, and skin, and other tissues. And this is a publication before it became public in a peer-reviewed paper. There was a preprint, and Karen Yitzhak is the first author, Gat Getz the last author, people from the Broad Institute in Boston in the US, where they did these many samples, 6,700 samples across 29 normal tissues with this pipeline that they call RNA mutek. Where are these mutations and how much of them they find? Well, they're not present everywhere. There are cases like glioblastoma where they can barely find anything. It looks all normal, and then it'll turn into a cancer that will end up, it may end up killing that individual. Other cases, they find them in other proportions. And then the allele fraction is not that high. You can see here the allele fraction of these mutations is from 0, but starting to go high at 0.2, 0.4, and 0.5 is already decreased quite massively, right? So they are not that pervasive, and hence they'll be difficult to measure. What about their profiles? What about their mutational signatures? They also change, and they make sense to the change of what has been found before, which is smoking causes mutations, and it causes a type of mutation that is different from other mutations. For example, UV has an effect on certain parts of the DNA, and it causes C to T deamination, as you can see here, which is also found in other patterns like aging, but also mutations of some genes that are correcting for these events like ApoBec, poly, and MSI. And so by looking at different tissues, you can measure how much of this is happening, which tissues are fully healthy, which others have some mutations, and the allele frequency at which they're found. And here the difficulty is that some of these allele frequencies are really minute. They're really, really low. And hence, we're going to need a very high precision method to detect them. So where do we go from here? Well, they say that it is not yet possible to properly assess probabilistically how far a tissue is from developing cancer. Meaning we can find mutations, and then in other individuals that have cancer, we can match some of the same mutations, but there's a gap in the middle between finding individuals that have some cells with some mutations and being to tell them when or if they will ever develop cancer. And this approach should also account for the dynamics of mutational clones by properly assessing normal endogenous mutational processes, the microenvironment of a tissue, exogenous exposures that are potentially avoidable, and the inheritance, meaning we all inherit some mutations or some variants that may affect the incidence of this. The environment, very important. The tissue microenvironment is also very important. All this complicates matters at the end when we want to translate these mutations we find in our body to a cancer prediction. And so to conclude, based on these results that we found, that they found recently in this paper, the Broad Institute and related, this is why early prediction of cancer is going to be hard. And this is Ewan Burney, co-director of the European Bioinformatics Institute in Cambridge, is saying, we all have or will have cancer or precancerous cells. The question is not, do these cells exist? For example, measuring them via cell-free DNA, but it is our body adequately managing the cancers it has got? And so it's a complex system-wide question. I hope you enjoyed this episode of the podcast. My Twitter handle, twitter.com/Alberto. I hope that you also follow me on LinkedIn and ask me questions. I hope to reply to the questions that you have. Follow this channel if you like these episodes, and a final reminder that this slide deck is available upon request. Until the next episode, goodbye.